Yeah, I can hear you. My preference is I, I, I cannot speak, Spanish, Portuguese.
So good day, everyone. I think we are ready to start.
Yes.
I see that we are now (21 participants connected). So I think we are ready. We are going to have simultaneous translation, and you have to set this language translation. I see, Mr. Picado, you are raising your hand. You would like to ask any question? We cannot hear you. So I think we can start. So welcome everyone to this webinar. I'm going to make just a very brief introduction of the current project and this triangular cooperation project. So I will share my screen. Please, if you have any problem, you can write in the chat, or if you have any question, you can also write the question in the chat. And before starting, I particularly would like to welcome some participants from TALIRI, researchers from TALIRI, also researchers from KALRO, but also some organizations like ABI from Switzerland and Savannah Forever and Tafori from Tanzania.
Hello, can you hear me now? Hello. Yeah. We have another participant. This is my daughter.
Please, I ask you to mute your mic.
Yeah, so this is my daughter. It's another participant today. Hello. Do you see me?
I don't know who's talking.
Yeah.
Okay, please try to have your mics off so we can hear us better. So just a brief introduction to this project. That is a triangular cooperation between Argentina, Kenya, Tanzania, and Germany. The name of the project is Strengthening Capacity Building to Improve the Livestock System of Smallholder Farming and Contribute to Agri-Food Sustainability in Kenya and Tanzania. This project is implemented by the Regional Fund for Triangular Cooperation. Oh, let's
What are we
see.
doing?
It's possible that, please.
To me a link.
So as I was saying, this is a project that is under the Regional Fund for Triangular Cooperation with partners from Latin America and the Caribbean. And in this case, is implemented with partners from Kenya and from Tanzania. But first of all, I would like to give you some highlights regarding triangular cooperation. That is a modality that is innovative and it has acquired increasing importance in recent years. This triangular cooperation is characterized by the participation of 3 different partners. One is the soliciting partner that have a necessity or a demand, a specific demand. The other is the principal partner who has a capacity to share or experience to share. And the third role is the role of a facilitator. In this case, is Germany that facilitates the exchange among soliciting and principal partners. So that's the thing we are doing from Germany with the GIZ. We are supporting the exchanges, in this case, between Argentina and Tanzania and Kenya. So this kind of project offers flexibility and also try to adapt specific solutions to particular contexts, and also try to strengthen cooperation between partners that perhaps don't usually work together, including private sector, civil society, scientific actors. By now, the Regional Fund has been promoting 142 triangular projects with 39 countries since 2011. But we also have other components, not only the project, but also we promote the concept of triangular cooperation. We try to develop and to diffuse the idea of triangular cooperation. We promote political dialogue. We have we make conferences every 2 years. In the last one, last year, also this project was presented by Halima and Kari there in Brazil, in the conference of Brazil. We also try with this program to strengthen cooperation ecosystems in the countries, and particularly we promote capacity development regarding how to manage these kinds of projects. So you can find much more information in our website. And now regarding the project background, regarding this project, the background has to do with bilateral technical cooperation between Argentina and Kenya. That's focus on improvement of livestock production in family farming in the arid and semi-arid lands of Kenya. The partners were KALRO from Kenya and INTA from Argentina. Some 4 missions were carried out to Kenya and 2 from Kenya to Argentina. But in this framework, contact with TALIRI was made, and they show interest in joining a new project. So they started political and institutional dialogue, and the proposal was prepared with the German and Argentine embassies, the Ministry of Agriculture in Kenya and Tanzania, with GIZ in Kenya and Tanzania also. So this is the background of the project, and the objective of the project has to do with some problems that were identified. They limit the limited forage supply, the pasture improvement, the advanced erosion project, the oversizing, invasive species without forage relevance, the implementation of inappropriate practice for the management of the pastures, lack of planning in the use of pastures, improper enclosure that modify ancestral practice of watershed use. So with all these problems identified, the objective of the project is to strengthen the technical capacity necessary to increase meat and milk production in the livestock system of smallholder farming. And the project has three strategies to achieve this purpose. One is increasing and stabilizing the forage supply. The second is facilitating technology incorporation into the production system and to promote better interaction between research, system extensions for sustainable rural development, and the designing and implementation of appropriate agricultural public policies. These are the target groups: researchers, extension workers, farmers, but also we have also indirect beneficiaries of the project that has to do with livestock farmers, and also students from different Kenian and Tanzanian universities that participate in the project, that are engaged, for example, in this kind of activities like this webinar, or that they participated in the workshop that we were carried out when experts from INTA visited Tanzania and Kenya. So we have these two components that has to do with the design and implementation of two experimental demonstration plots for forage management. We have advanced with one in Kenya, and we are moving forward with the second one in Tanzania. And the second component has to do with enhance the comprehensive management of livestock systems. Probably we are going to prepare workshops online for this component. And we see if we can do also training in Argentina or other steps by now, where we have done one mission to Argentina with two professionals from KALRO and two from TALIRI. They visited arid lands here in Argentina and see some technologies, and they have seen some technologies. And one mission to Kenya and Tanzania from INTA experts that also carried out some workshops in Kenya and in Tanzania. And as I said, we are moving forward with the implementation of these demonstration plots. So that is my introduction to this webinar. So to give you some general information about the project, but I now leave you in the hands of the Argentine experts. So Fernando, you have the floor.
Thank you very much, Alejandra. Well, now I will talk something about this webinar and why the objective of our demo plots in Kenya and Tanzania. I will share my screen. Wait a moment, please. Are you watching my screen now?
Yes, Fernando.
Yes.
Thank you. Well, first
Fernando,
of all, yes.
Just a second. I would like to check if Gonzalo can record. Is recording. I think there is no record ongoing. So Gonzalo or Paula.
I think that the record is ongoing because
yes,
there is
I know.
the red point is on, and we have 45
sorry.
minutes of recording. Well, first of all, good morning for all of you in Argentina, and good afternoon for all of you that are participating in this meeting from Kenya and Tanzania. It's a pleasure to to be able to talk to you and to receive you in this webinar in the frame of this trilateral cooperation project. I will talk about the challenges in livestock production systems in the arid and semi-arid lands of Kenya and Tanzania. Alejandra clearly introduced us to the problems that we have identified along our activities in the arid and semi-arid lands for the livestock production. You already know now the objectives of our project and the components. In one of these components, we have the commitment to implement demo plots in Kenya and Tanzania. Why? Well, I will try to explain why. This is the second project, the cooperation project that we are running from Argentina to Kenya, and now with Tanzania. We started our activities in 2017 with a cooperation project from the Argentinian Fund for South-South and Trilateral Cooperation. This first project was between Argentina and Kenya, and the objective was to improve livestock production in the arid and semi-arid zones. Now we are, during this project, we receive a lot of information, we search a lot of information, and then we submitted another project to the German Cooperation Agency. This project started in 2023, and this is linking another country, which is Tanzania also. So we are working with Kenya, Tanzania, Argentina, and of course, with the German Cooperation Agency. The strategy has always been a win-win strategy in which we try to learn from other partners for their experience, and we put our knowledge available for them to learn from us too. During these projects, one of the objectives of the first project was to identify and analyze the main restrictions that the livestock production systems in the arid and semi-arid lands were facing. For this, one of the activities was a participatory workshop. The first was in Isiolo, in Kenya, in 2019. In this workshop, we worked with a methodology of working in groups, with a broad participation of extensionists, researchers, and also policy makers, analyzing with a brainstorming technique the main restriction or problems or constraints that the livestock systems are facing in the arid and semi-arid lands in Kenya. And last year, just now in the frame of the new project, with this GIZ project, we carried out another workshop. This was in Arusha, in Tanzania. And in this case, we have been working with the methodology of a foresight analysis, identifying a desired situation for the livestock systems in the future, and then analyzing the restrictions that these systems are facing in the current situation. Both analyses show us that there are a lot of similarities between the arid and semi-arid zones in Kenya, Tanzania, and also in Argentina. These common and shared restrictions in the workshop in Isiolo, we organize them in topics. Some are related to water, another with extension service and research and innovation. Another restrictions are linked in the topic of pastures and livestock management, and there are other restrictions. Analyzing the pastures and livestock management restrictions are just the same that Alejandra has introduced us in the project. They are land degradation, overgrazing, high stocking rates, bush encroachment, the non-implementation of grazing management plans, and the low pasture seed production. In the Arusha workshop last year, we organized in productive topics, environmental topics, and social topics that are restrictions or problems. In the productive topics, we also found a decrease in forage supply, unproductive land, low production of pastures and pasture seeds, lack of water, few improved animal breeds, inadequate application of technologies, reduction of grazing land, overgrazing, and the increased importance of invasive plants, and increasing in animal diseases. Why we propose to work with demonstration plots? Well, firstly, to identify and evaluate on-site the performance of appropriate technological proposals to improve forage supply. Why? To improve forage supply is one of the main objectives, because a lot of restrictions or problems that we have found not only in the workshops, participatory workshops, but also in the visit to farmers and also talking with researchers and extensionists. This is one of the main needs to improve forage supply. And to improve forage supply, it's necessary to have available appropriate technological proposals. And technological proposals are appropriate when they are identified, analyzed, and built, constructed with the participation of the ones who will need to apply these technologies, so farmers. So we need to have a dialogue with farmers, with researchers, and with extensionists, just to be able to develop appropriate technologies. And this dialogue is going to happen in these demo plots. So in these demo plots, we will, and we are able to put to work these technological proposals, and then to analyze them with the active participation of farmers, extensionists, policymakers, and also the researchers. Another objective is to increase the capacity of actors linked in livestock production. These demo plots will work also as training areas with these technologies, just showing the development of these technologies and how to implement these technologies. And also another objective is to redesign the technological proposals by integrating lessons learned and contributions from both scientific and tacit knowledge. Scientific is the faster and deeply analyzed knowledge, but tacit knowledge has been developed along the years, and it is very robust, so it is necessary a dialogue between these different sources of knowledge. That's why we propose to work with these experimental demonstration plots, and they are already working. You have now some pictures of the demo plots in Kenia and Tanzania. Well, thank you very much. Muchas gracias. Asanteni.
Thank you very much, Fernando. We will continue with this program with Jonas Kisima. Jonas, please, if you can share your screen while I introduce himself. Jonas Kisima, he's research scientist. He's a specialist of rangeland science, pasture agronomy. He's working for Tanzania Livestock Research Institute. The short name is TALIRI, and he's working in the station in Tanga, in Tanzania. Jonas, could you share your screen? Okay. We are thinking that you can zoom it.
Can you see me?
Yes.
Yes. Good morning in Argentina and good afternoon in Tanzania. I hope you can see my presentation. As Zana introduced me, I thank every presenter who started. And my name is Jonas Kisima, based in Tanga, working for Tanzania Livestock Research Institute. And here I'm going to share with you the experiences on research trials and community engagement on how we can do rangeland restoration strategies in Tanzania, as we started previously from other collaborators, other projects, but we are also working with the Kabi Tafori as introduced by Alexandra. As you can see, as I said, TALIRI is working in Tanzania mainland, and as you can see from the map, we have 7 agroecological zones where Tanzania Livestock Research Institute conducts their research activities in 7 zones. You can see Lake Zone, Western Zone, Central part, Southern Zone, Northern part. I'm based in Eastern Zone, and we also have the Southern Zone. But we also have the semi-arid conditions, mainly in central part of Tanzania, Central Zone, but also in the Northern Zone and partly in the Eastern Zone of Tanzania. To start with my topic, I'll introduce that the livestock farming in Tanzania is an economic, but also the livelihood activity which supports the traditional way of farming, but it's also very critical because it supplies food and it serves the nutritional security to the populations in most parts of Africa, especially North, Southern, West, and Eastern. But this factor applies to Tanzania mainland, where traditional livestock farming almost carries the 95% of the traditional livestock farming, but also 5% of the modern type of farming. If you go to the livestock sector in Tanzania, 50% of all households keep livestock. They can keep poultry, goats, sheep, and what, but 50% almost of the population keep livestock. And this livestock contributes 30% of the agricultural GDP, but takes 7% of the total GDP of the country. And this is the source of protein from milk, meat, and eggs. And the sector plays a nutritional security component, income, and livelihood, as I said, but the sector also provides manure for crop production, and this makes life to sustainable agriculture. In many areas of Tanzania, farming operates below the optimum production potential due to some challenges, of which, if you go in deep, you will find the unimproved genetics of the animals, but also we face the challenges of water, land scarcity, conflicts, and also diseases, as mentioned by Fernando, but also the feed shortage, especially during the dry season, which of low quality and the quantity, the small quantities, which all of these challenges lead to low livestock productivity. If we go to specifically for the forages in Tanzania, we really see the shortage of forage supply, especially during the dry season, and this can take about 26% of the demand of the forage in Tanzania when it comes to the dry season, but also seeds or the forages, we can estimate the demand of 22,954 tons per year, but we can only produce 11.4 tons per year. So you can see this difference makes a very big demand of forage seeds. But we are now seeing in the rangeland restoration strategies the rising demand of native forage species, which are adapted to the areas in the semi-arid conditions. So this demand is not yet established as how much is demanded, but we see this demand during our restoration, rangeland restoration strategies. As we can see, the livestock farming in Tanzania is almost operated in 3 agroecological zones, which in humid agroecological zones where rainfall is very reliable, and this is where we find the the indoor system of livestock farming. But also we have the sub-humid agroecological zone with the moderate rains where traditional livestock farming is done, and semi-arid and arid conditions with very low rainfall, ranging from 450 to 750 millimeters of rainfall. So this is very low, and in most parts is livestock farming, especially traditional farming, is being operated in semi-arid areas. But we still see the opportunities in the sector where high demand of forage during the dry is very high. So whoever or investors who can venture in this can find a very big opportunity in this, or researchers, but also high demand of improved forage seeds for those who are now intensifying their livestock farming. With a high demand of drought-tolerant native forage seeds for the strategy of rangeland restoration activities. But we also see the high diversity in the wild, the native and local varieties, which can be taken for the improvement practices. Our research experience is in rangeland restoration as TALIRI. The efforts in addressing has been with the partners of development partners, International Livestock Research Institute, the KABI, TAFORI, and BioVision Africa. All these ongoing activities are giving us a very big experience in rangeland restoration. As you can see, as mentioned before, degraded rangelands is the area which, in semi-arid conditions, where the dependence of livestock in semi-arid conditions is very high. But you can see the photo in the photo. You can see the existing condition during the dry season. Very dry, bare land, no forages, but animals are still grazing in the same area. But also during the wet season, we still have a challenge of the invasive species. As you can see, it is very green, but lily, these species which are dominated in the grazing land are the invasive species which are not palatable, but also we find a bare land in the areas. The approaches we do with our experience in rangeland restoration, in most cases, we start with meeting with district authorities, also introduce the project, but also we have to select the villages within the districts. In this case, for our experience, Siha district, we selected Tingatinga village. We also selected Mawasiliano in the Longido district in the regions of Kilimanjaro in the northern part of Tanzania and Arusha. But all this, we have to meet the village leaders so that we can introduce whatever we want with the strategy of engaging the communities, so that whatever we are doing is really touching the communities, and also to see the willingness of the communities we are dealing with in the rangeland restoration strategy. So whenever we approach them, we have to meet the farmers to correct and share their knowledges. The farmers are very aware in their perspective in their indigenous area. So indigenous knowledge is very important in capturing what you can also do, but also incorporating what they know. Selection of research sites in the villages where we set up our demonstration plots is an approach, but we also visit the grazing lands, the degraded grazing lands, to see what we can do physically on the ground so that we don't go beyond from what they want. But we also visit the areas to identify available use forests and forage native species in the rangelands. But sometimes we engage the women groups to support us in the project to collect the native species. The interventions we use in our approach of rangeland restoration, in most cases we have to fence the area to demonstrate on how we can restrict animals from touching the areas we want to restore. And here we use the locally available materials for especially the cut-thorn trees, the sisal, fencing wires, euphobia. But in most cases, we see whatever is affordable to them. But we also use the intervention of partial bush clearing and also oversowing, using the species from outside the environment, but which can be compatible in the area, but also reseeding when we have the native species available in the area. But another intervention, we use the semicircular moon shape to retain water during the growing season, but also deferring the land at least for 2 growing seasons so that we can see the results. You can see the photos which are involved, where we involve the communities in Handeni in the previous activities, so that from the beginning up to the end of the project, the communities are well engaged so that they know exactly what we are doing. Even if the project ends, at least they can do something. You can also see with the native project, Native Plant for Environment and People, with the KABI International Organization who are working together. They are also implementing activities in Isiolo, Kenya. You can see the communities in Kenya are engaged in the activities to see whatever has been done on the ground for their cooperation. You can also see this photo where we are engaging women in Siha district in Kilimanjaro, how we can correct native species so that they can be used in the restoration strategies. But also during the rangeland restoration activities, we do engage the families with their own grazing lands, not the communal, but also the individuals' grazing lands, to see how we can engage the family labor to improve their own nearby home grazing land. You can see that photo with their kids in their family, but our staff is also teaching them how to plant and how to fence. All those are the strategies to engage the community at individual farm level. But also when we go to the communal lands, as you can see, it's very bushy, and sometimes the growing, the understory species cannot grow well. So we have to teach them on how to do partial bush clearing, so that whatever we plant under the trees, at least it can grow when you reduce the number of trees as an intervention. But for those who can afford it, also in the project areas, we use the barbed wire to fence the areas and see how this bare land can be improved with the oversowing or reseeding. But with the individual farms, we do use the sisal as a live fence to support restricting the animals to the grazing land. Those are the areas which were bare, but trying to show people how we can do rangeland restoration. You can see now in the start of the growing season, you can see something is coming up in the rinds where we disturb the land and plant something, at least to see how we can get the vegetation. If you restrict for 1 to 2 seasons, at least you can get something. Those are the areas like we fenced, but the green you see is the invasive species. But the fenced area, you can see the very good biomass where we teach them, if you restrict the land, at least you can get a very good vegetation, and you can suppress the invasive species in the fenced area because the palatable species will start to reappear. And this is the data, at least we can show with the area where we have fenced. At least you can see getting at least 5 tons to above where it was even very, very below the 3 tons per hectare, or sometimes in the area you find nothing. You can see the histograms of the forages we used. At least the Chloris Guyana showed the highest, and the other species, of course, because of time, I cannot go to the details, but at least we see the interventions we use in the oversowing. They show up from the bare land to something productive in the land. But in the approach of rangeland restoration, we have to teach the communities on how they can tie using the hand tools, the wooden boxes to make sure they can produce hay for their animals. They're taking at home from the plots we use. With this presentation, I would like to acknowledge or to thank the Minister of Livestock and Fisheries who are supporting the TALIRI Institute, but also the international community with the KABI, TAFORI, BioVision Africa, CETRAD, and the Nairobi University, whom we are working together in this strategy of rangeland restoration. So we thank everybody, and we welcome others to partner with us for the rangeland restoration strategies. With this presentation, I would like to thank the INTA and GIZ for supporting this webinar so that we can share our experiences and everything. Thank you very much for paying attention. Thank you.
Thank you very much, Jonas, for your presentation. It was a very clear and comprehensive overview of the work being done in Tanzania with local communities. Thank you. Now I will present to Bosco Kidake. Bosco, are you here? Okay. Bosco Kidake, he's a research scientist. He is working at the Kenian Agricultural and Livestock Research Organization. This is Kenya's main research organization in agriculture. He is working with pastures and livestock research as well as climate change issues. Okay, thank you, Jonas. Thank you, Bosco. Could you share your screen, please?
Is it visible?
Yes, we can see plant count.
Okay, let me just make it full.
Okay.
Yeah, good. I think it's good morning in Argentina, and for my colleagues in Tanzania and Kenya, good afternoon. I think good afternoon. Sorry, I came in late a little bit, but I want to share some few details of the work we've been doing around in the project, and plus some little work that involves evaluating forage and pasture seed plots. This is at some concerning the project, the Triangular Cooperation project, where we were involved in establishing demonstration plots, as highlighted by Fernando. So there's some metrics we've been measuring in the project, but before that, I'd like to just highlight the importance of the livestock sector in Kenya, where it contributes substantially to the gross domestic product of the country, and overall about 42% of the agriculture GDP. And actually, a large population relies on livestock in this part of the world, where 50% of it, and when you consider the pastoral population alone, we are having a situation of over 6 million of Kenyans. The country has a population of about 55 million. So a good percentage of about almost 20%, I mean, almost 5% relies heavily on the agriculture and the livestock sector. So our key products are beef, milk, sheep, goats, camels. There are other livestock that we keep, but for now, that is the one which substantially contributes greatly to the livelihoods of the population. Of course, the production systems, we have varied production systems, but majorly we have intensive, semi-intensive, and extensive systems. In this particular context, we are having farmers who are also small scale and also large scale. These large scale, they're not many, they're few, but they keep their animals majorly on grasses. These ones I'm talking about, semi-intensive and intensive, they are somehow also undertaking what we call zero grazing. And the intensive ones, they are highly specialized. Then they especially just undertake zero grazing as an activity that's a production system. Now, when it comes to forage production, because that's what our webinar was all about, all of us know that livestock majorly relies on grasses for the nutrition. And sometimes we have to establish grasses and also cultivate grasses to enhance feed availability. But above all, in the extensive systems, we have what we call natural pastures. These are grasses found in the wild. So livestock majorly rely on it in those areas where they graze opportunistically, walking around the bushes and the farms, having their nutrition. But above all, there is need for proper management of these particular resources to enhance livestock production and also maintain environmental services. So our project, the Triangular Cooperation Project, was majorly about fodder production. So we're working towards improving the forage resource base. But for us, in this case, being in research, we are also supposed to develop technologies that are aimed at enhancing fodder production. So as a result, like KALRO itself, being the Kenya's premier agricultural research organization, we've been involved in development of grass varieties. And these varieties are the ones which we take them to farmers. But one of the major shortcomings also that KALRO deals with is the quality of seeds, because pasture production relies on seeds. Seeds have been a problem. Only a few are available in the country. And this one includes Panicum maximum, Brachyria species, and also Floris Guyana. Some of these are imported a lot from South America and the United States. This includes the Brachyria and the Panicum maximum, which I know most of them come from your region in South America. And most of them are available in agrovets. But indigenous grasses like Cenchrus ciliaris, they are not totally available. So KALRO right now is involved in at least making sure that we grow these grasses, multiply them, commercialize, and even distribute to farmers for the purpose of rangeland reseeding and also pasture establishment. If you see that photo I've put there, that's part of our areas where we are doing some seed cleaning, because the sector itself is still in the infant stage. It's not developed well like the major commercial fishes. Now, we did establish some demonstration plots. I think this one has been highlighted before, and these demonstration plots were supposed to highlight technologies and also learn and teach farmers and students and other people what forage management is all about for increased livestock production. So we had an objective of evaluating different forage management practices at these particular demo plots. And the other one was also about seed production, but then of the objective that we are dealing with also number 2, we also had to do some evaluating bush and invasive species control practices for increased forage productivity. Now, just as a background, if you look at that particular slide, we established different range classes, and these photos I took from the specific plots that we've been working with at KALRO KIBOKO, the ones I established earlier in the year through support from the GIZ project and our organization. We had 4 grass species. One of them is called buffalo grass. The other one is bush rye, Maasai love grass, and the photos you can see the ones I've put there. We also had horsetail grass. Horsetail grass is the one that is in the lower right corner. We also established legume grasses because we wanted to also show the same, that livestock does not need grass alone. These are photos I also took from some of the plots where we had lucerne. We had another legume called butterfly pea. We had another one called cilantro. The scientific names are in brackets. These are very important species of legumes which are locally available, and we're trying to promote them because they are very good suppliers of protein to livestock. Now, when it comes to this particular plot, as a photo of the demo plot, which we established. Right now, it doesn't look like this. It's long since I was at the station. I've been away doing some work. This is a photo I got, I think it's about a month ago, from the same demo plot. It's a big plot of different varieties of grasses that we've been working with. But we've been doing a lot of work in those particular species. The same applies to this particular slide, whereby we are trying to evaluate different bush and invasive species control practices. Because a critical issue facing the drylands, one of them is bush encroachment and invasive species. These are limiting access to quality feeds, livestock, reducing forage production, decreasing carrying capacity, habitat alteration. Is also having economic impacts, which is a threat for livelihoods. The photo you can see is one of our colleagues, is the one who is moderating the webinar. When she visited Kenya, he was trying to get through one of the bushes, which is the one we try to manage in the project. Now, we've been employing different methodologies because the webinar was about how I understood it. We wanted to share amongst ourselves how we've been doing this work in terms of methodologies of relating this particular species. We employ different. Of course, it depends with the objective that we are doing. So for us, we established these bush grasses and at the same time try to go into a bush that we wanted to manage and showcase how to increase forage productivity. But as a scientist, of course, our technical staff, we were doing some measurements and some other data collection in these particular plots. One of them is what I'm calling seedling emergence. Of course, seedling emergence, we were doing some viability tests using different approaches in the lab. The seeds that we planted, we took them to the lab so that we ascertain the quality, so that we can have a good establishment. Prior to that, once you plant the grasses, of course, we had to go to the field and see how well the grasses are established. In the plot, of course, you can see on the right there is myself and my colleague where we were doing some recording of how the seedlings of the different grasses have emerged. We record this, of course, this is the data we definitely use later on in other approaches. Of course, forage structure. Grasses have got different parts, and when we want to do evaluate grasses, we have to look at different parts of the grass, some of them including leaves, heights, tiller measurements, stem and leaf parameters, the ones that are actually labeled in the photo on the right. So we did a lot of measurements, as you can see, where we were involved in evaluating different structures of the plant so that we can be able to compare with the different other species. And this information is normally very crucial for other management practices when you want to manage forages or grasses. We also did some morphological measurements. This is the species you are seeing there is one of the leguminous species we call Clotularia species, and we do some measurements just to ascertain growth parameters at different particular times and see how they are performing. And over time, the season, this gives us critical times where you can decide to know when the grass is performing well, when it needs what time of management, if the seeds are ready. So we do a lot of evaluation practices. Now, in terms of the vegetation itself, we do some plant counts at the same time. And this one is to help assessing the health of an established plot, because once you know how many grasses or how much the crop is in the field, it helps you measure and know if you are having adequate materials from the field. So for the bush activities, we are establishing what we call transects, laid across, and we lay quadrats along the transect so that we can be able to do some plant counts and also get some more information, such as density, numbers of the plants which are there, diversity, the different types of species that are in that particular environment, like especially in the bush plots, where we were collecting a lot of information in terms of the diversity of species. After doing the collection, the clearing, what is the response of the clearing? How much is coming out and which species and how frequent are these grasses coming up? Now, the grasses, I know range grasses don't have a lot of pest and disease instances, but some of these work as researchers, the technical staff, we do collect information regarding like how much pests and diseases are being visible. And as you can see on the photos, those are just some of the photos we are taking and seeing what are the incidences this quantitative information helps us come up with practices that ensure the crop is well managed and forage production is sustained over time. Furthermore, I think as we move on, we were doing a lot of biomass measurements over time. Actually, I was the first session has been about establishment, but like starting last week, because I clipped these materials the other week, from now until maybe going forward with whatever we have, we'll be able to assess the quantity of the grasses at different stages now. And this is normally done through quadrants, as I said before. But because sometimes grasses are normally utilized at different stages. So we need to know if a person wants to do livestock production, at what stage is the optimum time to get material enough to do some livestock production. You can also feed it as hay. So we harvest these and store it at the end of the season. So we know when to harvest. Is it the right time or not? So we do measurements at this particular time. As I mentioned, cutting is, of course, at different stages to determine productivity. And these are just some of the photos where we have people in the fields cutting the plots and doing a lot of measurements in the same plots that we established under the project, so that we can be able to estimate production and make use of that information to advise farmers or interested parties. Of course, it cannot end there. We look at forage quality analysis, and quality analysis is all about determining the composition of the chemical attributes or the nutritional attributes of this particular species. And the important ones, they are very many, but the important ones are about crude protein, minerals, ether, which are the parts, fiber components, and digestibility. And these ones, we do them under standard laboratory procedures. So far, I have just collected the materials. I intend to take them to the lab shortly to get some of the information so that when we write the technical report of the project, we can have a clear picture of what materials we were having. And then other measurements, of course, I mentioned earlier in the last presentation we had that we established trees also, these particular forage plots, demo plots we are planting. So we do shrub measurements, tree and shrub measurements, where we do the crown structure or the shrub structure itself and the 3 dimensions to be able to estimate. We use these, when you have this information, you can be able to measure even how much material, if you have browsing animals, they can consume from these. And also, from an ecological perspective, you can be able to measure carbon stocks in this particular environment. We do ground cover estimations, of course, using quadrants, and also other observations. Sometimes you just observe these plots. When you have a healthy ecosystem, you'll see other small animals, rodents moving around. So these observations sometimes we do make. So finally, we don't just do it for the sake of collecting data. The methodologies we employ, we do that also to train students and train farmers, and also take this knowledge outside, whereby we do a lot of farmer training activities in terms of social responsibility also, and also agricultural shows. So I think other than that, we also have a situation whereby we are doing some soil quality stuff. I'll be collecting soil samples over time, because from the time we started, then up to the end of the need, there are differences, changes in soil parameters, and these include soil physical parameters, including soil bulk density, texture, soil moisture, carbon nitrogen ratio, phosphorus, microbial, those are chemical properties, and then also microbial aspects, whereby we're looking at the different microbial properties under the different grasses or in our bushes, compared to areas which are not improved. Also microbial diversity, abundance. So we sample at different depths and also collect samples for laboratory processing, because you want to see how our soils are changing. Thanks a lot. Because of time, I didn't have time to go through all these particular details about each of those methodologies. But at least I think I've shared with what we are doing in the demo plot and what KALRO does. Thanks a lot for your attention.
Thank you very much, Bosco. It's a great job. It's a great work that you carry on in Kenya and KALRO. Now, together with Patricia Sabino, we will present our work. After this presentation, we have time for the questions and answer. I'm going to share my screen. Could you see it?
Yes, Roxana, we are watching your screens.
Okay, thank you. Well, with Patricia, we work in, we are both researchers at INTA.
Sorry, Roxana, you
Yes.
you have to put in presentation mode.
Oh, yes. I'm sorry. Is it okay?
No, you are still
No, it's not
with
okay.
the. You need to choose the another window.
Okay.
Fíjate, Roxana, si tenés abierta la presentación en tu computadora, ponela en modo presentación y selecciona esa ventana.
Is it okay now?
Yes, it is okay now.
Okay, thank you. Okay, with Patricia, we work in. Okay, thank you. We work in INTA in Santiago del Estero. INTA is the National Institute of Agricultural Technology. And I begin the presentation, and then Patricio will continue with the section on soil and analysis. I'm Roxana Ledesma. I am agronomist engineer, and my expertise is in drainage management and silvopastoral systems. Our presentation is titled Methods for Evaluating Vegetation and Soil Characteristics in cattle system in the Chaco region, Argentina. Here we have a map of Argentina, which is located in the southern part of South America. We live and work in the northern part of the country in a region known as the Chaco. The Chaco region shows a clear gradient in rainfall. In the western part, annual precipitation range from 800 to 100 millimeters, more or less. As we move to the east, the rainfall decreases significantly down to about 100 to 400 or 500 millimeters per year. The typical vegetation in these regions are forest and grasslands, as you can see in the photos on the right side. One of the main limitations in livestock production system in the Chaco region is the low forage productivity. This is largely due to sugar encroachment, as extensive areas of rangeland have been transformed into dense thickets and secondary forest. Sugar encroachment reduces the accessibility of forage and restricts the movement of both livestock and herders. As a result, the current stocking rates are low, sometimes very low. In our work, we apply the ecological site approach. This model classifies the landscape into different sites, each with similar characteristics, mainly in terms of topography, soil, and vegetation. On the left, you can see an aerial photo showing a mosaic of forest and grassland. On the right, there is a diagram illustrating two main ecological site types: highlands, are typically with forest and lowland, which are characterized by savannas and grasslands. In the satellite image, on the left of the screen, we identified 3 ecological site types: highlands, ecotone, and lowlands. To classify these areas, we use digital image processing tools. Within each ecological site, we collect field data using transit lines. Usually, we use 3 transect per ecological site. Along this transect, we measure vegetation parameters for grasses, shrubs, and trees, as well as various or different soil attributes that Patricio will show you. To assess grass productivity, we mainly use 2 methods. The first is the botanal methods, which stands for botanical analysis. It is simplified, indirect, or double sampling technique that is not destructive. And we use this method for large areas, for example, more than 20 hectares. This method allows us to estimate species composition, pasture yield, and palatability. It also provides information on ground cover, the amount of litter, plant growth, phases, feces of the animals, or the grazing intensity and other attributes. It's a very complete method for us. The second method we use is the cut and weigh method. It involves harvesting biomass and weighing the samples directly. This is a simple and direct approach, usually applied to small areas, less than 20 hectares. To evaluate forest structure, we use methods specifically designed for assessing shrubs and trees. Among the techniques we use are the T-square sampling procedure, or sometimes it's called nearest neighbor, the point quarter method, and others depending on the context on the vegetation. In the diagram shown here, you can see how the point quarter method is applied. Along each transect established within an ecological site, we mark a specific sample point. At each point, we measure the nearest individual trees or shrubs in each of the 4 quadrants. We measure indicators such as species abundance and richness, canopy cover, plant height, volume, number of sprouts. As you can see in this picture, this is these are the sprouts of a typical tree in Chaco region. It's named Quebracho Blanco. Stem diameter at breast height, as you can see in this photo, where a student is making this work. In this photo, you can see different situations of shrub encroachment, a major issue in rangeland. Shrub encroachment significantly affects paddocks accessibility, so one of our goals is to estimate shrub volume, because shrub volume affects paddock accessibility. So one of our goals is to estimate shrub volume. The greater the shrub volume, the lower the accessibility for livestock. In the field, we measure the crown diameter and height of individual shrubs. With these measurements and basic formulas, we calculate shrub volume. From this data, we can also estimate above ground biomass using allometric equation. You can see the the other Conti, he has some equation for this calculation. This information is highly relevant for climate change studies as it allows us to estimate the carbon stock of each silvopastoral system or pastoral system. Well, in our field work with farmers and rangers, we apply the adaptive management approach. This approach begins by identifying key indicators in each range and in each ecological site level. We collect data on vegetation, soil, and socioeconomic variables to establish a baseline. It means a starting point for long-term monitoring. Once the baseline is established, we monitor these indicators and design appropriate management practice. This process follows a cycle. You can see the words plan, after do, or apply, adjust, and evaluate. If we observe that certain practices are having negative impacts on the soil, vegetation, grasses, or forest structure, we dialogue with the farmer to adjust this practice. Then we reevaluate, new data, analyze the results, and repeat the cycle. This continuous feedback loop allows for ongoing improvement and sustainability of management practice. Okay, now Patricio will continue with the soil attributes. I remember you, Patricio will speak in Spanish. If you need the translation, you can go to the 3 points in the bar and select the language, English, the translation English to Spanish. Well, Patricio, you share
Hola, saludos
your
a
screen.
la audiencia. Bueno, mi nombre es Patricio Sabino, soy investigador de INTA y especialista en manejo de suelos. Roxana, ¿vos vas a poder pasarme las diapositivas?
Ah, sí, pero I'm sorry. ¿Está?
Bien.
Mm.
Bueno, la idea de hoy es mostrar un poco algo del trabajo que venimos realizando en diferentes sitios de evaluación, tratando de realizar muestreo de indicadores edáficos en sistemas ganaderos bajo monte nativo o bosque nativo. Pasa la Rosa. La siguiente, Rosa. Los objetivos de los trabajos en general siguen la idea de caracterizar primero en principio una línea de base o un punto de partida en base a características físicas y químicas del suelo mediante un conjunto de indicadores que se seleccionan. Posteriormente, y en base a esos indicadores, detectar los cambios que son generados por un cambio de uso o un cambio de manejo en esos suelos que se relacionen con estos parámetros edáficos. Pasa la Roxana. Bien, los indicadores con los que trabajamos son variados y inician en base a un set de indicadores de calidad que es grande y del cual luego comenzamos a seleccionar cuáles son los más específicos o mejores para nuestros suelos, ¿sí? O para el suelo en cuestión. En este caso, los suelos en general son aplustoles torreorténticos, con características de textura franco-arcillosas. Hay que aclarar que estos suelos no se desarrollan in situ, sino que son formados a partir de depósitos eólicos o aluviales, y tienen una gran profundidad, no tienen problemas de profundidad para exploración radicular, y altos contenidos de fósforo en el material que le dan origen, como así también altos contenidos de sales solubles en la matriz del suelo. Por lo tanto, uno de los parámetros que tenemos en cuenta en general es el fósforo extraíble o el fósforo disponible, siendo que este nutriente es un macronutriente y tiende a tener un ciclo biogeoquímico en donde no se repone naturalmente. Por lo tanto, los procesos degradativos, cuando se consume pasto y se extrae ese fósforo que tiene el pasto, tiende a generar una caída de los valores de este indicador. Por lo tanto, es un valor a ir monitoreando para toma de decisiones posteriores. La materia orgánica, por otro lado, también es un indicador crítico para estos suelos, debido a que el contenido de materia orgánica naturalmente es muy bajo. Esto se debe a la alta tasa de mineralización y descomposición que tienen estos suelos, ya que las altas temperaturas de este clima semiárido generan un rápido consumo de la materia orgánica del suelo cuando se pasa de un monte nativo o de un bosque nativo a agricultura. Se estima que aproximadamente la pérdida de materia orgánica de estos suelos es aproximadamente de un 50% en 6 años. Por otro lado, junto con el fósforo y la materia orgánica, también se monitoreó o se monitorea el pH del suelo, ya que es una gran influencia al momento de dejar disponible los nutrientes. Hay nutrientes que son pH dependientes, por lo tanto, por ejemplo, el fósforo, y es un atributo que es importante monitorear. Tanto la materia orgánica como el fósforo y el pH se los monitorea en profundidades de 0,20 centímetros y de 20 a 40 centímetros. Como se comentó anteriormente, estos suelos también tienen un alto contenido de sales, por lo tanto se monitorea la conductividad eléctrica para hacer relación a la salinidad que tiene el suelo, y también la densidad aparente es otro indicador que se toma en cuenta, debido al pisoteo que generan los animales sobre la superficie y la pérdida de porosidad que le imprimen al sistema cuando se pone en producción estos ambientes. Finalmente, se analiza también la textura del suelo, ya que permite inferir otros indicadores o otras características del suelo, como es la capacidad de intercambio catiónico, la porosidad y la retención de agua, y tanto la conductividad eléctrica como la textura y la densidad aparente se monitorean en las profundidades de 0,20, 20,40 y 40,60, dependiendo de si son muestras simples o compuestas. Ahora voy a explicar cuestiones de muestreo simple o compuesto, porque de eso dependen las características, los indicadores, cómo se los van a muestrear. Pásalo, Rox, por favor. Bien, el monitoreo de variables como la materia orgánica, el pH, el fósforo extraíble, se lo hacen muestras simples. Estas muestras se toman a lo largo de una transecta de 100 metros que tienen 5 metros de ancho aproximadamente. Las transectas coinciden o son coincidentes con los sectores de recolección de muestras de pastura. Las muestras se comienzan a tomar sobre la transecta a los 30 metros del inicio de la transecta, es decir, que el inicio, que coincide normalmente con un alambre o con un punto de acceso, no se toma muestra debido a que está muy perturbado, muy disturbado. Las muestras compuestas, como la palabra lo indica, se componen de 3 submuestras cada 30 metros, o sea, 30, 60 y 90, y en 20 centímetros y 40 centímetros. Es decir, de la transecta saldremos con solamente 2 muestras que tienen 3 submuestras internas de 0 a los 30, a los 60 y a los 90, y otra de 20 a 40 que tiene las submuestras de 30, 60 y 90. Por otro lado, la muestra simple está compuesta por una sola muestra, que se la toma en el centro de la transecta, es decir, en los 60 metros de transecta. Esta muestra solamente contiene suelo de ese sector y se toma de 0,20, 20,40 y 40,60 y normalmente se la utiliza para determinaciones de conductividad eléctrica, de densidad aparente y generalmente se sale de todo, de cada una, o sea, de la transecta con las muestras que no tienen que superar más de 1 kilogramo de material para analizar. Pasa la Roxana, por favor. Bien, la recolección de muestras. Para la recolección de muestras utilizamos equipos particulares, principalmente por la dureza que tienen nuestros suelos. Nuestros suelos tienden a ser muy duros para introducir caladores. Entonces usamos un barreno de densidad aparente, que es el que vemos en la imagen de la derecha. Con la densidad aparente nos permitirá luego hacer cálculos para peso de capa arable y posteriormente calcular la cantidad de nutrientes, kilogramos de nutrientes que tenemos. Para las muestras de profundidad, tanto simples como compuestas, utilizamos un barreno helicoidal, que es el que tenemos en la imagen del frente, del centro, perdón, pero previo al muestreo se retira el horizonte o horizonte orgánico para que éste no genere ruido al momento de tomar las muestras, ¿sí? Solamente se toman muestras de los horizontes minerales. Pásalo, Rox, por favor. Bien. Hay un pequeño detalle con el muestreo que les acabo de explicar. El tema es que, más allá de haber hecho una ambientación por sitio ecológico, nosotros tenemos características en los sistemas productivos que tienen sectores que tienen cobertura arbórea y otros sectores que no tienen cobertura arbórea. Estos sectores, cada uno de estos sectores, presentan características de evapotranspiración y mineralización diferentes. Por lo tanto, como son sistemas complejos, tratamos de abarcar toda la variabilidad y que las muestras obtenidas del campo sean representativas de lo que sucede realmente. Para eso tomamos, por cada submuestra que les había explicado antes, en la muestra compuesta y en la muestra simple, tomamos una submuestra, es decir, cada punto a los 30 metros, por ejemplo, tomaremos una submuestra debajo de la copa del árbol y una submuestra fuera de la copa del árbol, a fin de poder compensar la variación que hay en estos dos sectores de todo el ambiente, ¿sí? Pásalo, Roxana, por favor. Bien, Por último, el monitoreo de estos indicadores de suelo se realiza en principio sobre una línea de base en donde sacamos materia orgánica, fósforo extraíble, conductividad eléctrica, densidad aparente y textura. Y posteriormente esa línea de base, que la línea de base sin que se le haya hecho ninguna intervención al lote, posteriormente, luego de haber hecho la intervención, se hacen determinaciones de conductividad eléctrica, pH, fósforo extraíble, materia orgánica y densidad aparente en las épocas de invierno, que es cuando los procesos son más lentos debido a las bajas temperaturas. Por otro lado, la textura del suelo se la toma una sola vez, solamente en la línea de base, debido a que la textura es una variable de poca variabilidad temporal. Con todos estos datos, lo que logramos nosotros es ir monitoreando y posteriormente tomar decisiones de cómo ir manejando estos sistemas para poder lograr la sustentabilidad tanto del sistema pastoril como del sistema suelo y lograr los mejores rendimientos en cantidad de producción de carne, que es lo que realmente uno trata de lograr con esto. Si hay cambios importantes en los indicadores edáficos, normalmente se toman decisiones de manejo en conjunto con el equipo técnico para poder subsanar dichos problemas. Pásalo, Rox. Bueno, muchas gracias por la oportunidad de presentar el trabajo.
Gracias, Patricio. Thank you very much, Patricio, for your presentation. I will send the word to Alejandra because she needs to explain a little thing.
That's very brief before you start with the questions. I see there are some very interesting ones in the chat. I put in the chat a link for a form. It's a link to a survey that help us to evaluate and to improve and to plan next steps. So I ask you, please, there are very few questions that if you can answer, it will be very useful for us as the project. Thank you.
Well, now we start the part of the questions and answers. First of all, thank you very much, Roxana, Patricio, Jonas, and Bosco for your presentations. It has been very rich, and it was possible for us to see the similarities and differences between the different productive livestock production areas in Kenya, Tanzania, and Argentina. We have some questions for some are specifically for one of the presenters, and others are for all the presenters. I will start with the common questions for all the presenters. The first is, how are the work they have been presented contribute to carbon sequestrations? Any plans to include the carbon footprint in the study sites? Can they be registered for carbon trade credit? Well, you tell me who will answer first this question. Who would like to start answering this question? Maybe Roxana, Bosco, or Jonas, Patricio?
Maybe I can answer this question in relation with the studies and the work that we have here in Argentina, and maybe it's a possibility to do this in the plots in Kenya and Tanzania. when we collect the data of biomass of grasses and shrubs, for example, we can take a data for the carbon sequestration, the stock of carbon. With Patricio also, he is studying the organic matter, and also Bosco showed in his presentation that this data is collected too, and maybe this is a source of information for the carbon storage.
Thank you very much, Roxana. Jonas, Bosco, something to add with this question? Hello, Jonas, are you listening to me? Not at the moment. Thank you, Bosco. Jonas, you want to add something?
Yes. Okay. With our project with the native plans for environment and people, where we are working with Tafori, we are now we started the forage component, but we are now going to the forest component. So the Tafori, Tanzania Forest Research Institute, they are now thinking on how to organize the communities for restoration of the forest gardens. And that is the point where we'll be able to, to capture the quantification of the carbon sequestration. And we are thinking whether if the forests will be, the community forest will be okay, then the carbon business will be involved in the project. Thank you.
Thank you very much, Jonas. I will go ahead with another question for all the presenters. It is, most range grasses are adapted to the different soil types. So how do you improve different varieties of grasses for production? Is it clear? Maybe Roxana, you can start answering this question, or Jonas or Bosco.
Could you repeat, please?
Yes.
Yes.
The question is, most range grasses are adapted to the different soil type. So how do you improve different varieties of grasses for production?
Maybe it's maybe can Patricio answer this question because it's related with the soil types. For example, here in our region, we have some sites that in which the conductivity, electric conductivity is high, and in these cases, we usually see a pasture, Grama Rhodes. It's a pasture that is tolerant to salinity, and this is the case in our region.
Thank you, Roxana. Maybe Bosco, Jonas, something to add to this, or Patricio, I don't know.
Si quiero, yo, para agregar un poco a lo que comenta Roxana, más allá de la tolerancia que tienen ciertas forrajeras a la salinidad, como es el problema en nuestros ensayos, hay que comenzar a pensar en cómo manejar el sistema suelo-planta. ¿Por qué? Porque cuando los procesos de evaporación superan a los procesos de transpiración de las plantas, se genera un arrastre de sales a la superficie del suelo, impidiendo posteriormente una germinación de las semillas que uno incorpora para el sitio a poner en producción. esta salinización de la superficie impide la germinación de las pasturas, independientemente de su tolerancia, y entra el sistema en un círculo vicioso en donde la oferta forrajera se reduce no en sí por la potencialidad de la planta, sino por una limitación edáfica, lo que hace que el manejo deje de tener sentido hacerlo desde una visión pura y exclusivamente del pasto o de la forrajera, sino hay que verlo en conjunto con el suelo. En nuestro caso, la reducción de los pastoreos o la reducción de la biomasa de ese pasto la reducción del pastoreo genera que las sales comiencen a irse a profundidad y permiten una mejor producción del pasto que está establecido y posterior una mejor germinación de las mismas semillas de ese pasto, ¿sí? Por lo tanto, mejor oferta forrajera.
Thank you, Patricio. Well, we will continue with another question for all the presenters. This is: a single grass or legume species may have many accessions or varieties, and these varieties may be different. So, how are you choosing to work with it? Is it clear?
Yes. I think if you allow me to answer that.
Thank you, Bosco. Go ahead.
Yes, for instance, in Kenya here, one of the jobs, one of the activities we are working with at KALRO is evaluating different grass accessions. So we take these particular grasses, we collect them from wherever they are, because there are many of them are conserved at the National Gene Bank. Come and plant them at the station, and we start what we call selection by just observing how they are performing. Then we look at their performance. If there are 10 particular accessions, we look at like the 5 best performing ones and take them to the next stage again of evaluation. And this depends with the objective, because sometimes the objective might be biomass production. So we look at which one is producing more biomass, and then we start working with that one. So for instance, the grasses that I've been presenting about, if you allow me to look at this particular, let me just try and share my screen.
Yes. Of course.
Yes, this particular photo I'm showing here. The grass in front is the same species. I mean, it's an accession of this one which you are seeing ahead, like 2 steps ahead. And you can see there are 2 different colors, but they belong to the same variety, which is Cenchrus ciliaris. So these are 2 accessions that we selected from a pool of about 30 accessions that we got from the National Gene Bank, simply because their biomass production was high. So we decided to work on these particular 2 to increase production and start multiplying so that we can have them as released as varieties, new varieties in the country. So we work them through breeding by selection. That's one of the ways we do that. Thank you.
Thank you, Bosco.
The same works for legumes.
Well,
Yes.
thank you very much, Bosco.
Okay.
Well, there are now something to add, Jonas or Roxana or Patricio.
I think Bosco has no comment.
Well, thank you very much. So we continue with the.
So as usual, I think
Now I have a question
this
specifically
one
for Jonas.
will be led by
Uh,
Augustine.
So I can see we are
Sorry.
very busy.
Oh, that's me.
uh,
So how was the day? What have you achieved?
Sorry, please to mute your microphones if you are not going to speak. Well, This question is specifically for Jonas. It is. I will read it. I was curious about the process you followed when organizing women's group to collect native species for their regeneration strategies. Did you conduct interviews or any kind of participatory activity to identify which native species were most used or known by local communities? Or were they given a predefined list of species to collect? I am particularly interested in how local knowledge was incorporated in the selection process. Well, Jonas, is it clear, the question for you?
Yes, Fernando, it is clear. Thank you.
Well, thank you.
Okay, thank you. During the engagement of the community, we were conducting meetings at different stages. So when we started visiting the grazing areas, but that was not restricted only for women. We were also considering all the villagers who attended the meetings. So we had to go together in the grazing lands, but also in there are areas called Olalili. They conserve the lands during the dry season for the grazing of the sick animals nearby homesteads. They can be the individual or the communal. So what we did, it was to go with the meeting participants in the areas and show them what has been restricted, which has grown. So we listed with the participants, what do you think is the best species which are preferred by the animals? So we were listing the list from the grazing land, which is restricted which has matured. But another approach was during the rainy season, when we started now the trials, the demo plots. We were asking the meeting participants to go nearby the bush when the plants have started growing. So we said we want the good species, the vegetative part, you bring them so that we can plant in our demo plot. So they brought about two grasses, because in most parts they are overgrazed, so that you cannot find the good plants. But when they have started growing, they can go to the bushes and uproot the nice plants, and they come with them, and they show the meeting participants. So those were the two approaches, bringing them to us, but also going together in the bush and identify and list them together. Thank you.
Thank you, Jonas. Well, I will continue with the questions. There is one question specifically for Bosco. The question is, what type of soil microbial properties data collected and its impacts with the study objective? Did you catch the question? Bosco, are you still there here?
Okay, you're asking what type?
It is what type of soil microbial properties data collected and its impacts with the study objective?
Yeah, as I mentioned earlier, right now we've not yet done any of that work. We just collected the soil samples, and we are planning to do that soil microbial aspects later on, like in the course of the coming months and the next 1 month, because the plots we just managed to establish them recently. But in terms of whatever specifics, that I cannot be able to answer them right now, because that's one field of work I've not been really involved in deeply in terms of microbial aspects. I work a lot on the above ground aspects, but concerning the below ground in terms of microbial attributes, that one I will not be comfortable answering right now. Maybe one of my colleagues who, Roxana, maybe you can assist me on that.
Roxana, do you have something to add?
No, really no, because we don't use this type of biomass, microbial biomass in the soil.
Well, thank you, Roxana. Thank you, Bosco. I have another question. One is for you, Bosco, and it is, how long does it take to restore degraded lands, and how do you reseeding process?
Well, it depends. Degraded lands, it depends on severity of degradation, first of all. If an area is very severely degraded, definitely it will take longer to bring it back, but depends also with the interventions that you put into these activities. There are areas, from experience, there are areas which have been able to recover within one season, depending on factors such as soils, climate of the area, rainfall, and even the kinds of species you put into that particular area in terms of restoration or rehabilitation. So it can take as long as one season, other areas it can take long, because areas which are most degraded, especially in the drylands, they normally have more challenges than one. It is not just the degradation itself, because it has to recover back to at a certain point, and bringing it back the area of which it was very productive, it can take as long as even 10 years. But it varies, depends with how severe it is, depends with also the management interventions you put in. Jonas talked about maybe just leaving areas and using twigs and making the place without accepting animals to get into such areas. That such areas recover quickly. But areas where there are livestock maybe crisscrossing, it becomes very difficult. I'm working with communities right now. I'm in the field somewhere where we are doing some restoration activities, and they are coming up with strategies to accelerate restoration. They were telling me, if you just leave an area for 2 seasons, 2 good rain seasons, that means it's just about 1 year. It normally recovers very quickly. Then the reseeding component, how we do it, of course, sometimes you just go to an area and do broadcasting, and some other areas you have to go and disturb the soil a little bit when it is very bare, so that the grasses that you put in there, they're not washed up by other insects or wind or water. So you disturb the soil a little bit by using maybe mechanical means, equipment, machinery, and plant your grasses in such environments. Thank you.
Thank you, Bosco. Well, I will go ahead with 3 more questions because it is necessary 5 minutes for the closing of the webinar, and there is very little time. So the next question is for Jonas, and it is, you conducted bush clearing. How effective it was? Is total bush clearing recommended in arid lands like those in Tanzania? And how did you manage the seed bank to control regrowth? Is it clear, this question for you, Jonas?
For the bush clearing is clear, but for the seed bank control regrowth is not clear. Maybe I start. Yes, I can start with the bush clearing. Because most of the grazing lands, some are bushy, wooded grasslands or whatever. So it depends on the nature of the grazing land. But in most cases, when you find a community grazing, and they say this is the grazing land, you have to assess how bushy the grazing land is. Sometimes there are other areas where you find it's too bushy, where they don't allow the understory species which are edible to animals. They are just trees, more trees. So when we engage the community, we ask them to reduce the number of trees which are not necessarily useful, because in most cases, the grazing lands are used for many purposes. They are for medicinal, for firewood, for shade. So it depends on the community use of the trees. So in most cases, we advise the farmers, and we ask them, which trees do you want to reduce? So the reduction of trees, depending on the plant density, we reduce depending on their preferences. But the essence is to reduce the number of trees so that we allow the understory species which are edible to animals accessible even for the animals. So it was not clear to me, did you manage the seed bank to control regrowth? Maybe I will answer if somebody can chat again. Okay. There are areas where you find they are very bare land, where no vegetation completely. We are now trying to take the soil samples to see the soil seedbed, what is available in the soil, even if the land is very bare. But you can take the samples and assess which can germinate from the area. But otherwise, in some areas, when we have restricted by fencing, at least we allow the vegetation to mature, seed maturity, so that they can drop down for regrowth in the next season. So it depends on how the vegetation cover is, but the maturity of seeds or the way to establish from the vegetative part or from the seeds. Maybe that is what I can comment. Thank you.
Well, thank you, Jonas. Well, we have more questions, but we have no more time. So some questions that will be left, and we hope to continue organizing these kind of webinars to be able to interact with our partners in Kenya and Tanzania. So now for the closing remarks, I will invite again Roxana for some words as a closing remarks of this webinar. Please, Roxana.
Okay. Yes. Well, we are very happy to make this event, this webinar. Thank you very much for all the presenters. Thanks for Patricio. Patricio is in his holidays, but he is now here, and thank you very much. Thank you, Jonas and Bosco. We are working with them some years before, and well, no more. Thank you very much for all the participants in this webinar.
Well, thank you very much, Roxana. I think that this is an enriching process, and we are building this cooperation between Tanzania, Argentina, and Kenya. We found a lot of similarities between our livestock production systems. So we think this strategy of win-win, in which we learn from each other, is working very well. This is one of the experiences in this webinar in which we can see that we are learning from each other. So thank you very much for all the presenters, for all the participants. I think that Cynthia Melville from the Argentinian Embassy something. So go ahead, Cynthia, please.
Thank you, Fernando. Thank you very much. My name is Cynthia Melville. I'm Deputy Head of Mission of the Embassy of Argentina in Kenya. We are also a concurrent embassy with Tanzania. And I have had the pleasure of meeting most of you last November in your last mission to Kenya and Tanzania. And I'm very delighted to see you all again in this webinar, especially want to thank the presenters, very interesting presentations. I just wish to make two very short remarks. First, I would like to express how pleased we are to see the great progress that has been achieved in the cooperation project and the significant work that has been undertaken in the demo plots. It is indeed very impressive. And this is thanks to the hard work and the commitment of the whole team, of all the partners involved. INTA, TALIRI, KALRO, KIBOKO as facilitating partners. You make a great team, and I will use your own jargon and your own metaphors, and the high yield results we are seeing today are indeed a testament to your excellent teamwork. And a second remark, very short, if you will allow me, this from a diplomatic standpoint. you must be very aware that Argentina is very, very proud to be part of this South-South Regional Cooperation Project. We seek to make a significant contribution to livestock management systems by increasing forage supply in arid and semi-arid areas, and we also seek to promote food security. But what we value most in this project is that it offers the possibility of building bridges, of sharing technical knowledge in the farming sector, which is a sector that is very important to 3 countries. And it also enables us to strengthen our ties with Kenya and Tanzania and having a real impact on the communities and engaging with them, as Dr. Kisima and Dr. Kidake mentioned in their presentations. And lastly, I'd like to convey the message that our embassy in Kenya stands ready to support you in the next stage of the mission, the project, and obviously looks forward to meeting you again here in Nairobi or elsewhere in Kenya and Tanzania. Thank you very much.
Well, thank you very much, Cynthia. We have some hand raised, but there is no more time, so we hope to continue in touch in other webinars and in other missions to be able to see you personally. Thank you very much. Thank you very much, Cynthia, Alejandra, Roxana, Bosco, Patricio, and Jonas, and all the participants. Thank you very much. See you. Bye-bye.
Bye-bye.
Bye.
Bye.
Gracias. Bye. Thank you.
Bye.
Thank you very much, everybody.
Thank you.